Learn biology, DNA, genes, and chromosomes. Introduction to heredity and the molecule of inheritance. We begin our study tonight at the very beginning of the biological question. As you settle in, let your mind turn to the most fundamental observation of the living world. We will explore the concept of heredity. This is the ancient, observable truth that like begets like. It is the simple, profound fact that offspring resemble their parents, whether in a field of wheat, a family of wolves, or a human family. For countless millennia, humanity has witnessed this phenomenon. We have built entire civilizations upon it, domesticating animals and cultivating crops by carefully selecting for desirable traits, all based on this predictable pattern of inheritance. Yet, for almost all of human history, the physical mechanism behind this inheritance remained a deep and profound mystery. It was a black box. We could see the results clearly, but the process itself, the how, was completely unknown. In this first lesson, we will begin our journey into that black box. We will gently open the lid and introduce the central molecule, the physical substance, responsible for carrying this immense legacy of information from one generation to the next. Let us now define our terms with clarity. Heredity is the formal name for this process, the transmission of traits from one generation to the next. The study of heredity is called genetics. The information that is being passed along is our genetic information. You can think of this information as a blueprint or a detailed instruction manual for building and operating a living organism. This blueprint determines the traits of that organism. A trait is any observable characteristic. Some traits are visible to the naked eye, such as your height, the color of your eyes, or the shape of a leaf. Other traits are microscopic, such as the specific type of proteins your body can build, or the chemical structure of your red blood cells. All of these instructions, from the grandest to the most minute, are contained within this genetic blueprint. The central question, then, is what is this blueprint? What is it made of? How can information be stored in a physical, chemical form? The answer, which eluded humanity for so long, lies in a molecule of breathtaking elegance and staggering information density. This molecule, the physical carrier of all our biological instructions, is called deoxyribonucleic acid, or, more simply, DNA. This is the molecule of inheritance. It is a long, chain-like molecule found within the cells of nearly every living organism on Earth. In complex organisms like us, called eukaryotes, this precious DNA is carefully protected within a central fortified compartment in the cell, known as the nucleus. Think of it as the master instruction manual for building and operating an organism, kept safe in the head office. It contains every chapter, every page, and every word required to guide the development of a single fertilized egg into a complex trillion-celled being. It is the thread that connects us to our most distant ancestors, a chemical archive of our entire evolutionary history. This molecule, DNA, is the primary subject of our study. To understand biology, we must first understand how this remarkable substance works. Let that concept settle in. The molecule of life is DNA. Today, this is a foundational fact, taught from an early age and accepted as central to our understanding of the world. But it is important to place ourselves in the minds of scientists from the past. This knowledge is very new. For the first half of the 20th century, the scientific community was engaged in a great and vigorous debate. What carried the code of life? The leading candidate was not DNA, it was protein. This was a perfectly logical, intelligent, and reasonable assumption. Proteins are the workhorses of the cell. They are the enzymes that drive chemical reactions, the structural components that give cells their shape, the motors that allow them to move, and the signals they use to communicate. Proteins do everything. Furthermore, proteins are molecules of immense complexity. They are built from a vocabulary of 20 different amino acids, which can be arranged in nearly infinite sequences and then folded into complex, beautiful, and functional three-dimensional shapes. Life, in all its staggering diversity, must be the product of an equally complex molecule. This was the prevailing logic. DNA, by contrast, was thought to be a stupid molecule. It was known to be built from only 4 repeating units, or letters. To the brilliant scientists of the time, it seemed far too simple, too monotonous, to possibly contain the rich, complex instructions for building a human being. It was believed to be a simple structural scaffold, perhaps a kind of biological glue, holding the real genetic material, the proteins, in place. This debate raged for decades. It was the central mystery of modern biology, the eventual discovery that the simpler, stupid molecule was, in fact, the true and only master code, was a complete revolution in thought. It proved that life's complexity arose not from the complexity of its components, but from the near-infinite arrangement of simple ones. Understanding DNA is therefore the key to understanding this fundamental and deeply elegant principle of all biology. This concept of DNA as the universal blueprint is not merely an abstract piece of trivia and useful only for passing an examination. It is the single most important organizing principle in all of modern biology. It is the foundation upon which entire fields are built, fields that have reshaped our world. The most profound implication is its universality. The fact that all living things on Earth, from the simplest, single-celled bacterium living in a hot spring to the complex ecosystems of a rainforest, to you, listening to this, all use the same molecule and the same fundamental code to store their information is the most powerful evidence we have for a single, common origin of life. This universality is what makes modern biotechnology possible. It is the reason we can study a gene in a fruit fly or a mouse to understand a similar gene in a human. It is how we can harness bacteria, giving them a piece of human DNA, and turn them into tiny factories to produce human proteins, such as life-saving insulin for diabetics. This concept is the bedrock of genetics. It is the core of modern medicine, as we now understand that countless diseases are, at their root, diseases of information, small errors, or typos in the DNA blueprint. And finally, it is the physical mechanism behind the grand theory of evolution. Evolution, in its simplest terms, is the process of change in this DNA blueprint over vast geological stretches of time as organisms adapt to their changing environments. We have now established what the molecule of inheritance is. We have named it deoxyribonucleic acid. We have also established why it was a mystery for so long, with proteins as the leading but ultimately incorrect hypothesis. The logical question that follows is, how did we come to know this? How was DNA definitively proven, against the prevailing scientific opinion, to be the true molecule of life? Our next lesson will explore the key, elegant experiments that uncovered this fundamental truth. The search for the genetic material. Welcome back. In our previous lesson, we established the central molecule of life, deoxyribonucleic acid, or DNA. We now accept this as a fundamental truth. But science is not a collection of truths received from on high. It is a process, a slow and methodical journey of discovery, often against the current of prevailing opinion. As we discussed, the scientific community of the early 20th century was largely convinced that complex proteins, not simple DNA, must be the carriers of genetic information. Therefore, our task in this lesson is to journey back in time. We will trace the story of how this scientific assumption was overturned. We will follow the thread of evidence, from a mysterious observation in a laboratory to a series of elegant experiments that, piece by piece, built an irrefutable case. This segment covers the crucial experiments that prove DNA, and not protein, carries the code of life. Let your mind settle as we explore this great scientific detective story. Our story begins in 1928 with a British bacteriologist named Frederick Griffith. He was not trying to find the molecule of inheritance. He was studying Streptococcus pneumoniae, the bacterium responsible for pneumonia, hoping to develop a vaccine. Griffith was working with 2 different strains, or varieties, of this bacterium. The first strain was called the strain. When grown in a lab, it formed smooth, glistening colonies. This was because it had a protective outer capsule, which shielded it from the host's immune system. This strain was virulent when injected into a mouse. The mouse would develop pneumonia and die. The second strain was called the R strain. It formed rough-looking colonies because it lacked that protective capsule. Without this shield, the R strain was easily defeated by the immune system. It was non-virulent when injected into a mouse. The mouse remained perfectly healthy. Griffith performed a series of simple, logical experiments. First, as expected, he injected a mouse with living strain bacteria. The mouse died. Second, he injected another mouse with living R strain bacteria. The mouse lived. Third, he took the deadly strain bacteria and killed them with heat. He injected this heat-killed strain into a mouse. The mouse, as he predicted, lived. The bacteria were dead and could not cause infection. It was the fourth experiment that changed history. Griffith mixed two harmless substances, the heat-killed strain and the living R strain. On their own, neither could harm the mouse. But when injected together, the mouse developed pneumonia and died. This was a complete shock. When Griffith autopsied the mouse, he found its blood swarming with living, virulent, smooth-coated strain bacteria. Somehow, an active principle from the dead strain cells had been transferred to the living R strain cells, permanently changing them. This principle had given them the genetic instructions for making the protective capsule, transforming them from harmless R strain into deadly strain. Griffith called this unknown substance the transforming principle, but he did not know what it was. The search for this transforming principle began. For over a decade, scientists tried to purify it. Then, in 1944, a team of researchers at the Rockefeller Institute, Oswald Avery, Colin MacLeod, and Macklin McCarty, published a landmark paper. They had spent years methodically isolating Griffith's transforming principle. Their process was one of painstaking elimination. They started with a large batch of heat-killed strain bacteria and purified the transforming principle, confirming it could still change R strain into strain in a test tube. Then, they treated samples of this purified substance with different enzymes. First, they used proteases, enzymes that destroy proteins. If protein was the transforming principle, destroying it should stop the transformation. But it did not. The R strain still transformed. The principle was not protein. Next, they used RNases, enzymes that destroy RNA. Again, the transformation continued, unaffected. The principle was not RNA. Finally, they used DNases, enzymes that specifically destroy DNA. When they added this enzyme to their purified sample, the transformation stopped. The R strain bacteria remained harmlessly R strain. The conclusion was unavoidable. The transforming principle, the very substance of heredity, was DNA. This should have been the end of the story. But the scientific community remained deeply skeptical. The protein-first hypothesis was too deeply entrenched. Many argued that the DNA preparation used by Avery's team must have been contaminated with small, undetected amounts of protein, and that this contaminating protein was the real genetic material. The definitive proof, the final unambiguous experiment that silenced the debate, came in 1952. It was conducted by Alfred Hershey and Martha Chase. They chose a perfect organism for the job, a bacteriophage, which is a type of virus that infects bacteria. A phage is incredibly simple. It is little more than a protein coat surrounding a core of DNA. When it infects a bacterium, it attaches to the outside and injects its genetic material, leaving its empty coat behind. The injected material then hijacks the bacterium, forcing it to build thousands of new phages. Hershey and Chase designed an experiment of beautiful simplicity. They knew that protein contains the element sulfur, but DNA does not, and they knew that DNA contains the element phosphorus, but protein does not. They prepared two separate batches of bacteriophages. The first batch was grown in a medium containing radioactive sulfur 3,5. This made all the phage protein coats radioactive. The second batch was grown in a medium with radioactive phosphorus 3,2. This made all the phage DNA cores radioactive. They then took these two labeled batches and used them to infect two separate cultures of bacteria. After letting the phages attach and inject their genetic material, Hershey and Chase used a common kitchen blender to shear the empty phage coats from the surfaces of the bacteria. Finally, they used a centrifuge to separate the mixture. The heavier bacteria, containing the injected material, formed a pellet at the bottom, while the lighter, empty phage coats remained in the liquid supernatant above. The results were clear and conclusive. In the first batch, which used sulfur 3,5, the radioactivity was found in the supernatant with the empty protein coats. The protein had stayed outside. In the second batch, using phosphorus 3,2, the radioactivity was found in the pellet inside the bacteria. The DNA had gone in. This was the final word. The DNA, and only the DNA, had entered the bacteria and carried the genetic instructions to build the next generation of viruses. The case was closed. The Hershey-Chase experiment is a classic. It is celebrated not just for its conclusion, but for its elegant design. It perfectly separated the two candidates, protein and DNA, and cleanly tracked their fates. It bypassed the contamination arguments that had plagued the Avery experiment. It convinced the world because its logic was irrefutable. It demonstrated that even in the face of widespread, logical skepticism, a single, well-designed experiment can pivot the entire direction of science. This narrative from Griffith's accidental discovery to Avery's methodical chemistry to Hershey and Chase's elegant proof is more than just a history lesson. It is a perfect case study of the scientific method in action. It shows how a single, unexplained observation, Griffith, can blossom into a critical question. It shows how that question can be pursued with patient, rigorous, and systematic testing, Avery. And it shows how lingering skepticism drives the need for even more creative and definitive proof, Hershey-Chase, ultimately leading to a new and solid foundation of knowledge. Now that the scientific world was finally and definitively convinced that DNA was the molecule of life, a new, monumental question consumed the field. The race was on. If this simple molecule held the entire blueprint for life, what did it look like? What was its physical structure that could possibly allow it to store so much information and, just as importantly, to be copied from one generation to the next? Our next segment reveals the story of the double helix, the structure of deoxyribonucleic acid, DNA. In our previous lesson, we completed our search. We followed the trail of evidence that began with Griffith's transforming principle and culminated in the definitive Hershey-Chase experiment. We now know with certainty that deoxyribonucleic acid, or DNA, is the one true molecule of inheritance. This knowledge, however, immediately presents us with an even greater question. How? How can this single molecule perform its two profound and seemingly contradictory functions? First, how can it store the vast, intricate library of instructions needed to build a living organism? And second, how can it be copied, perfectly, millions of times, to be passed from cell to cell and from one generation to the next? The answer to this, how, is not philosophical. It is mechanical. It lies in the physical shape, the chemical composition, and the precise three-dimensional architecture of the DNA molecule itself. In this lesson, we will cease to treat DNA as a concept and begin to examine it as a physical object. We will explore its structure, piece by piece, and see how its form is perfectly and beautifully suited to its function. Let us begin with the most basic description. DNA is a polymer. This is a scientific term for a very simple idea. A polymer is a large molecule made by linking many smaller, repeating units together in a long chain. You can think of it like a train, built from many individual railway cars linked one after another, or a necklace, composed of many individual beads strung together. The individual repeating unit of DNA, the bead in our necklace, is called a nucleotide. This nucleotide is, itself, made of 3 smaller parts. If you are still awake, picture these 3 components in your mind. First, there is a sugar. In DNA, this specific sugar is called deoxyribose. It is a 5-carbon sugar, and it forms the core of the nucleotide. Second, there is a phosphate group. This is a cluster of phosphorus and oxygen atoms. It is this component that gives DNA the A in its name, acid. The phosphate groups are acidic and, critically, they carry a negative electrical charge, a fact that becomes very important when we consider how DNA interacts with other molecules. And third, and most importantly for information, there is a nitrogenous base. This is the information part of the nucleotide, the letter in the genetic alphabet. While the sugar and the phosphate are the same in every nucleotide, this nitrogenous base can be one of 4 different types. These are adenine, which we will call A, guanine, which we will call G, cytosine, which we will call C, and thymine, which we will call T. These 4 bases are the entire alphabet of the genetic code. So, we have a complete nucleotide, a sugar, a phosphate, and one of 4 bases. Now, how do they link together to form the polymer? The phosphate group of one nucleotide forms a strong chemical bond with the deoxyribose sugar of the next nucleotide. This creates an extremely strong, stable chain, linking nucleotide after nucleotide. This chain is known as the sugar-phosphate backbone. It is the string of our necklace, while the bases A, T, C, and G hang off to the side. But DNA is not just one long chain. This is the crucial point. DNA is a double helix. This means it is composed of 2 of these long polynucleotide strands, 2 long chains that are intimately connected to each other. These 2 strands spiral around an imaginary central axis, like 2 staircases winding around each other. How are these 2 strands held together? They are connected at their bases. The bases from one strand reach across and form chemical bonds with the bases from the opposite strand. These bonds are not the strong, permanent, covalent bonds that form the backbone. Instead, they are weaker, more delicate bonds called hydrogen bonds. And this connection is not random. It follows a strict and non-negotiable rule, the most important rule in all of genetics. It is called the base pairing rule. Adenine, A, on one strand, can only pair with thymine, T, on the other strand. They fit together perfectly, like a lock and key. Similarly, guanine, G, on one strand, can only pair with cytosine, C, on the other. A always pairs with T, G always pairs with C. This rule has a beautiful chemical logic. The bases are divided into 2 types. Adenine and guanine are larger, 2-ringed structures called purines. Cytosine and thymine are smaller, single-ringed structures called pyrimidines. The base pairing rule always pairs a large purine with a small pyrimidine, A with T, G with C. This elegant design ensures that the distance between the 2 backbones, the width of the molecule, remains perfectly uniform all the way down its entire length. The most common analogy for the double helix, and the most effective one, is that of a twisted ladder. Let this image form in your mind. The 2 long sides, or handrails, of this ladder are the sugar-phosphate backbones. These are the structural supports, strong and stable, protecting the information held within. The rungs of the ladder, the steps you would climb, are the paired nitrogenous bases. Each rung is one pair, either an A paired with a T or a G paired with a C. The rungs are flat and stack neatly on top of each other, providing further stability. And finally, the entire ladder is twisted into a gentle right-handed spiral. This is the helix. This shape is not arbitrary. It is the molecule's most stable, low-energy configuration, the natural way these chemical forces balance. There is one more layer of structural detail that is essential for understanding how DNA works. The 2 strands of the ladder are antiparallel. This simply means they run in opposite directions. Think of a two-way street. One strand runs in one direction, and the other strand runs in the exact opposite direction. Scientists label these directions based on the carbon atoms in the deoxyribose sugar. One strand is said to run from 5' to 3', while its partner strand runs from 3' to 5'. This antiparallel arrangement may seem like a minor detail, but as we will see in later lessons, it is absolutely critical. The cellular machinery that reads and copies DNA can only travel in one direction, so this structure dictates the entire mechanism of replication and transcription. Let us also reconsider the bonds. The backbone is held together by strong covalent bonds. This is like a permanent weld. The code on a single strand is secure and will not easily break. The two strands are held to each other by weaker hydrogen bonds. This is like a zipper. This dual bond system is a masterpiece of functional design. The strong bonds protect the code, while the weak bonds allow the two strands to be gently unzipped by cellular enzymes. This unzipping is the first step in both copying the DNA and reading its instructions. This structure, this double helix, is the key. Its form is its function. Let us connect the structure back to our original two questions. First, how does it store information? The information is stored in the sequence of the bases along one of the strands. The order of A's, T's, C's, and G's is a code. It is a language. A sequence like a TTCGG means something different from GGCTAA. Given that a DNA molecule can be millions or even billions of bases long, the amount of information that can be stored is, for all practical purposes, infinite. Second, how is it copied? The answer lies in the base pairing rule. Because A must pair with T and G must pair with C, the two strands are complementary. They are not identical, but they are perfect reflections of each other. If you unzip the ladder and are left with just one strand, for example, one that reads a GCT, you know, with absolute certainty what the other strand must be. It must be TCGA. This provides a simple, elegant, and almost perfect mechanism for replication. Each strand can serve as a template for building its missing partner. We have now examined the intricate and beautiful architecture of the DNA molecule. We have seen how its structure as an antiparallel, base-paired double helix is perfectly designed to store and to copy the blueprint of life. This discovery, piecing together the sugars, the phosphates, the bases, and the helical twist, was not a single flash of insight by one person. It was a complex, competitive, and brilliant intellectual race, built on the foundations of other scientists' work. Our next lesson will step away from the chemistry and tell the human story behind this discovery, honoring the key figures whose work and whose data piece together this beautiful puzzle. Key figures: Watson, Crick, Franklin, and Wilkins. In our last lesson, we explored the structure of DNA as a physical object. We learned about the double helix, the sugar-phosphate backbones, and the specific pairing of the nitrogenous bases. This structure is a masterpiece of chemical engineering, perfectly suited for its dual roles of storing information and self-replication. But this knowledge was not delivered to us complete. It was discovered. It was the prize in one of the greatest intellectual races of the 20th century, a story of brilliant minds, competing methodologies, and crucial moments of insight. Great scientific breakthroughs are never just about the science. They are complex human stories of collaboration, competition, ambition, and inspiration. In this segment, we will provide the historical context for the discovery of the double helix. We will honor the key figures whose combined work, whether in direct collaboration or in fierce competition, piece together this beautiful, life-defining puzzle. Our story takes place primarily in England, in the years immediately following World War II, and it centres on two rival laboratories. The first location is King's College, London. Here, the primary method of investigation was X-ray crystallography. This is a difficult, meticulous, and powerful technique. In essence, a scientist must first coax a molecule, like DNA, to form a pure, ordered crystal. Then, they shine a focused beam of X-rays at that crystal. As the X-rays pass through, they strike the atoms in the crystal and diffract or scatter in a specific pattern. This pattern, which looks like a complex arrangement of spots and smudges, is captured on photographic film. To the untrained eye, it is meaningless. But to a physicist or chemist, this diffraction pattern is a set of mathematical clues. By measuring the angles and intensities of these spots, one can work backward to deduce the three-dimensional structure of the molecule that created them. At King's College, two scientists were at the forefront of this work. The first was Maurice Wilkins, a New Zealand-born British physicist who had begun using X-ray diffraction to study DNA. He was later joined by Rosalind Franklin, a brilliant and fiercely independent chemist who was already a world-renowned expert in X-ray crystallography from her work on the structure of coal. Franklin was recruited to King's to lead the X-ray diffraction work on DNA. Her arrival, however, led to a strained and complex working relationship with Wilkins. The second location is the Cavendish Laboratory at Cambridge University. Here, a different approach was being taken by James Watson and Francis Crick. Watson was a very young American biologist, brash and singularly focused on discovering the structure of the gene. Crick was a British physicist, slightly older, known for his brilliant mind and infectious laugh. They were not experimentalists in the same way Franklin and Wilkins were. They were model builders. Their strategy was to take all the known data from other researchers, the established rules of chemistry, the dimensions of the component molecules, and any experimental data they could find and try to build a physical model, like a child's construction set, that fit all the facts. They believed that if they could build a model that was chemically stable and explained all the data, it would, by necessity, be the correct one. Their first attempts were failures, but they were in a race, and they knew that the team at King's College, particularly Rosalind Franklin, was producing the best experimental data in the world. Rosalind Franklin, through her meticulous and skillful work, had made an enormous breakthrough. She had discovered that DNA could exist in two different forms, which she named the A form and the B form, depending on the humidity in the chamber. The A form produced a complex X-ray pattern that was difficult to interpret, but the B form, the wetter form of DNA, produced a pattern that was strikingly clear. In May 1952, Franklin, working with her graduate student Raymond Gosling, captured a stunning X-ray diffraction image of the B form. This image, which would become known to history as Photo 51, was the single most important piece of evidence in the entire story. It was a clear, beautiful X shape, right in the middle of the diffraction pattern. To a trained crystallographer like Franklin, this X was not a mystery. It was the unmistakable mathematical signature of a helical structure. Furthermore, the spacing of the spots on the film provided precise, hard measurements. From this single photograph, Franklin was able to deduce that the helix had a specific pitch, that the sugar phosphate backbones must be on the outside of the molecule, not the inside, as many had assumed, and that the repeating base pairs were stacked on the inside, like rungs on a ladder. She was methodically, carefully, and correctly solving the problem. This is where the story becomes complex. In early 1953, Maurice Wilkins, whose relationship with Franklin had deteriorated, showed Photo 51 to James Watson. It is a matter of historical debate whether he did this with or without Franklin's full knowledge or permission. For Watson, seeing that X was a blinding flash of insight, it was the proof. He knew, instantly and unequivocally, that the structure was a helix. Armed with this critical confirmation, Watson returned to Cambridge. He and Crick now had another vital piece of the puzzle, a rule discovered by another scientist, Erwin Chargaff. Chargaff had analyzed the chemical composition of DNA from many different species and found that the amount of adenine, O, always equaled the amount of thymine, T, and the amount of guanine, G, always equaled the amount of cytosine, C. With Franklin's data confirming a helix of specific dimensions, and Chargaff's rules demanding an AT and GC pairing, Watson and Crick returned to their models. Their final brilliant insight was to pair the bases on the inside. They realized that pairing a two-ring purine, A or G, with a single-ring pyrimidine, T or C, would create rungs of a uniform width, perfectly matching the dimensions from Franklin's photo. Their model of a right-handed, anti-parallel double helix with the backbones on the outside and the AT-GC pairs on the inside was chemically stable and explained all the data. They had found it. In April 1953, the scientific journal Nature published a series of 3 articles. The first, and most famous, was the short, one-page paper by Watson and Crick proposing their double helix model. In the same issue, in support of this model, were 2 other papers, one by Wilkins and his colleagues, and one by Franklin and Gosling, presenting their experimental X-ray data. The discovery was a turning point for all of biology. It unlocked the door to the modern era of molecular biology, genetics, and biotechnology. In 1962, James Watson, Francis Crick, and Maurice Wilkins were jointly awarded the Nobel Prize in Physiology or Medicine for this discovery. There is a tragic and controversial postscript to this story. Rosalind Franklin was not, and could not be, awarded the prize. She had passed away in 1958 at the young age of 37 from ovarian cancer, likely linked to her extensive work with X-ray radiation. The Nobel Prize is never awarded posthumously. For many years, her foundational contribution was relegated to a footnote. Today, however, history and science have reached a clear consensus. While Watson and Crick brilliantly synthesized the data and built the correct model, it was Rosalind Franklin's meticulous, powerful experimental work, encapsulated in Photo 51, that provided the essential and non-negotiable data needed to unlock the secret of life. This story shows us that science is a profoundly human endeavor, a tapestry of insight, hard work, and complex relationships. The discovery of the double helix gave us the book of life. We now understand its physical structure, but a book is useless if you cannot read its language. Now that we understand the structure, we must learn to decipher the message written within it. Our next lesson explores the alphabet of this book, the genetic code. The genetic code and base pairing rules. In our previous lessons, we journeyed through the story of discovery. We now know, thanks to the collective work of Watson, Crick, Franklin, and Wilkins, that the structure of DNA is a double helix. We can picture it in our minds as a long, twisted ladder. This structure is the book of life. But a book, no matter how beautifully bound, is useless if you cannot read the language written inside. We have identified the physical object. We must now learn to decipher the message. This lesson focuses on the language of DNA. We will explore the set of rules by which information is stored on this molecule. This is the genetic code. We will learn the alphabet, the words, and the grammar that allow this simple, 4-letter molecule to write the complex and beautiful story of all living things. Let your mind be receptive as we learn to read this fundamental language. The first, and most foundational, rule we must revisit is the base pairing rule. As we learned, the rungs of the DNA ladder are formed by pairs of nitrogenous bases. Adenine A always pairs with thymine T. Guanine G always pairs with cytosine C. This rule is the key to DNA's stability and its ability to be copied. But it is also the key to its language. The information, the message itself, is written in the sequence of bases along one strand of the DNA. Think of one side of the ladder as the page we are reading. The other side of the ladder, its perfect complement, acts as a backup copy. Because A must pair with T and G with C, the sequence of one strand dictates the exact sequence of the other. This ensures the code is stored safely, with its own built-in mirror image. Now, let us look at that code itself. The language of DNA is an alphabet with only 4 letters, A, T, C, and G. That is all. The profound complexity of life is written with just these 4 characters. The next question scientists had to solve was, how are these letters formed into words? If the cell read one letter at a time, there would only be 4 possible words: A, T, C, G. This was not nearly enough, as cells use 20 different amino acids to build their proteins. Proteins are the machines and structures of the cell, and amino acids are their building blocks. 4-word vocabulary cannot possibly build a machine that requires 20 different parts. What if the code was read in 2-letter words? For example, AT, AG, TC, and so on. If you calculate the combinations, 4 letters taken 2 at a time give 16 possible words. 4x 4 equals 16. This is closer, but still not enough to specify all 20 amino acids. The solution, therefore, had to be a 3-letter code. The cell reads the DNA alphabet in groups of 3. This gives 64 possible combinations. 4x 4x 4 equals 64. This is more than enough to specify all 20 amino acids, and also to provide punctuation for the code. This 3-letter word in the DNA sequence is called a codon. A sequence of DNA, then, is a long sentence made of these 3-letter codons. For example, the sequence ATGTCAAGG is read by the cell as the words ATG, then TCA, then AG. Each of these codons means something specific. ATG might mean start building the protein here. TCA specifies a particular amino acid, perhaps serine. AGG specifies another, arginine. In this way, a long sequence of codons spells out the exact order in which amino acids should be linked together, like a recipe instructing a chef on which ingredients to add, and in what order. This chain of amino acids, once complete, folds into a complex 3-dimensional shape, becoming a functional protein, an enzyme, a muscle fiber, or a hormone. This recipe for one specific protein is what we call a gene. This genetic code has several remarkable properties. The first, and most profound, is its universality. The code is not just the human code or the tree code. It is the life code. The same codons specify the same amino acids in almost every single living organism on Earth, from the simplest bacterium to the largest blue whale. The codon GGC instructs the cell to add the amino acid glycine, whether that cell is in a blade of grass, in a fungus, or in your own body. This stunning uniformity is one of the most powerful pieces of evidence we have that all life on this planet descended from a single common ancestor, a distant, primordial cell that first discovered this language. The second property of the code is its degeneracy, or redundancy. This sounds complex, but it is a very simple and elegant concept. We have 64 possible codons, but only 20 amino acids to code for. What do the extra codons do? First, a few of them act as punctuation. There is one primary start codon in the messenger RNA, its ARG, which tells the cellular machinery, "This is the beginning of the recipe. Start translating here." And there are 3 different stop codons that signal, "This is the end of the recipe. The protein is complete." This still leaves many codons to specify just 20 amino acids. The solution is that the code is redundant. This means that multiple codons can code for the same amino acid. For example, the amino acid leucine is specified by 6 different codons: CUU, CUC, CUA, and CUG, all mean leucine. This redundancy is not a flaw, it is a critical safety feature. It makes the code robust and resistant to errors. Imagine a random mutation, a typo in the DNA, changes the last letter of a CUU codon to a CUC. The word has changed, but its meaning has not. The cell still reads leucine, the correct amino acid is added, and the final protein is perfectly functional. This wobble, in the third position of many codons, acts as a buffer, protecting our cells from the constant, small errors that can occur in the DNA. Finally, the code is non-overlapping and is read in a specific reading frame. The cell's machinery attaches to the DNA, or its RNA copy, at the start signal and reads 3 letters, then the next 3, then the next 3. It reads A, B, C, D, E, F, G, H, I. It does not read A, B, C, then B, C, D, then C, D, E. This ensures that the message is read in one clear, unambiguous way from start to finish. The ability to read and understand this code is the single most powerful tool in modern biology. It has completely transformed medicine, agriculture, and our understanding of history. Because the code is universal, we can practice genetic engineering. We can take the human gene for insulin, the recipe for the insulin protein, and insert it into the DNA of a simple bacterium. Because the bacterium reads the exact same language, it will read the human gene and begin producing human insulin, which we can then harvest and give to diabetics. This knowledge also allows us to understand, at the molecular level, the basis of genetic diseases. We can sequence a person's DNA, reading their code letter for letter. We can find the exact typo that causes a disease. For example, sickle cell anemia is caused by a single letter change in a single codon. GAG, which codes for glutamic acid, is mutated to GTG, which codes for valine. This one-letter change, in one word, in one recipe, changes the shape of the entire hemoglobin protein, with devastating effects. We have now learned the language of life. We understand its alphabet of 4 letters, its words of 3 letters called codons, and its grammar. We know how the information is stored. But before any cell can divide, before life can be passed from one generation to the next, this entire, vast genetic library, billions of letters long, must be copied, and copied perfectly. Our next lesson will explore the elegant molecular machinery that performs this incredible task, the process of DNA replication. DNA replication, the semi-conservative model. In our lessons so far, we have unlocked the physical form of the genetic blueprint. We understand the double helix, the base pairing rules, and the 4-letter language that writes the instructions for all of life. We have established that this book of life is stored safely in our cells. But storage is only half the story. The defining feature of life is its ability to perpetuate itself. A single cell divides into 2. A fertilized egg grows into a trillion-celled organism. For this to happen, the entire instruction manual, all 3 billion letters of the human genome, must be copied with near-perfect accuracy. Every new cell in your body, whether it is a skin cell, a liver cell, or a neuron, requires its own complete and identical copy of the original blueprint. The sheer scale of this task is difficult to comprehend. It is like hand-copying a library of thousands of books, letter for letter, in just a few hours, and making almost no mistakes. This vital process, one of the most fundamental acts of life, is called DNA replication. In this lesson, we will explore the elegant and efficient molecular machinery that carries out this essential task. The process begins with a profound question. When you copy a double-stranded molecule, what does the copy look like? Do you create an entirely new, fresh copy, leaving the original master molecule intact? Or do you use the original in some way? The answer was confirmed in 1958 by 2 scientists, Matthew Meselson and Franklin Stahl. Their work proved that DNA replication follows a semi-conservative model. The name itself is descriptive. Semi means half, and conservative means saved. In this model, each of the 2 strands in the original double helix serves as a template, or a guide, for building a new, complementary strand. Let this process unfold in your mind. It is a sequence of mechanical steps, a beautiful molecular dance choreographed by specialized proteins called enzymes. It begins at specific locations on the DNA, known as origins of replication. An initiator enzyme arrives and begins to pry the 2 strands apart. This creates a small bubble. Then, The main enzyme of unzipping arrives. It is called helicase. Its job is to move along the DNA, breaking the weak hydrogen bonds that hold the base pairs together. Imagine a zipper. The helicase is the pull tab, running down the length of the zipper, separating the two rows of teeth. As it moves, this unzipping creates a Y-shaped structure known as a replication fork. Now, the two original strands are separated and exposed. Each one is now a template. The sequence of bases on each strand, A, T, C, G, is bare, ready to be read. This is when the star of the show arrives. The master builder enzyme is called DNA polymerase. This remarkable protein has a singular vital job. It reads the template strand, one base at a time. As it reads, it grabs free-floating nucleotides from the rich soup inside the nucleus. And, following the non-negotiable base pairing rule, it adds the one, and only, correct complementary partner. If it reads an A on the template strand, it adds a T to the new strand. If it reads a G, it adds a C. It moves along the template, building a new, perfectly matched, and complementary strand. This happens on both of the original template strands at the same time. The replication fork moves down the line, and the two DNA polymerase enzymes follow behind it, one on each strand, building two new strands. The result is the completion of the process. Where there was once one double helix, there are now two. They are completely identical in their genetic sequence. And, critically, each of these two new molecules is a hybrid. Each one is composed of one old strand, the original template, and one new strand, freshly synthesized by the polymerase. This is the semi-conservative model. Half of the original molecule is saved or conserved in each of the two new copies. This model is beautiful in its simplicity, but the reality inside the cell has one more layer of profound complexity. This complexity arises from a detail we learned earlier. The two strands of the DNA ladder are anti-parallel. They run in opposite directions. Think of a two-lane highway. One lane runs north and the other runs south. The DNA polymerase enzyme, our master builder, is like a specialized car. It can only drive in one direction. It can only add new nucleotides to one specific end of the growing strand, the 3' end. This limitation creates a fascinating puzzle. For one of the template strands, this is not a problem. This is the leading strand. Its template is oriented in the correct direction. As the helicase unzips the DNA, this polymerase can drive along smoothly, building one long, continuous new strand. But what about the other strand? This is the lagging strand. Its template is facing the wrong way. The polymerase cannot drive smoothly down this highway. It would be driving against traffic. So, how does the cell solve this? It solves it by building the lagging strand in reverse, in small discontinuous pieces. As the helicase unzips a new section, the DNA polymerase jumps onto the lagging strand near the fork and builds a short fragment backwards, away from the fork, until it hits the previously made piece. As the fork unzips further, the polymerase must jump back again, closer to the fork, and synthesize another small fragment. These short, backward-stitched fragments of DNA are known as Okazaki fragments, named after the scientist who discovered them. The lagging strand is, therefore, not built in one smooth piece, but as a series of small stitched-together fragments. Finally, another enzyme, a molecular glue called DNA ligase, comes along and seals the gaps between these fragments, joining them into a single, complete strand. This entire process is not just elegant, it is also unbelievably fast and accurate. In humans, DNA polymerase can add about 50 new bases every second, and it has a proofreading function. After it adds a new base, it checks its work. If it has accidentally added the wrong base, an A opposite a G, for example, it can backspace, clip out the incorrect nucleotide, and insert the correct one. This proofreading function is so effective that DNA replication makes only about one error for every billion bases it copies. This process, This molecular dance of helicase, polymerase, and ligase is not a distant, abstract concept. It is the engine of your own life. It is happening in your body right now, billions upon billions of times. This is how you grow from a single fertilized egg to the complex adult you are. Every cell division was, and is, powered by DNA replication. This is how you heal. When you cut your skin or break a bone, it is this process that allows new cells to be created, copying the genetic blueprint to build new, healthy tissue. It is how you maintain your body. Cells in your digestive tract and your blood are constantly wearing out and being replaced. DNA replication ensures their replacements are perfect copies. The incredible accuracy of this process, its proofreading ability, is all that stands between order and chaos. When this system fails, even slightly, and an error is missed, it results in a mutation, a permanent change in the genetic code. Such changes can be the starting point for diseases. The fidelity of DNA replication is, in a very real sense, the guardian of your health. We have now seen how the blueprint of life is structured and how it is perfectly copied to ensure its continuity. We have a safe, stable library of instructions, and we have a perfect high-fidelity copying machine. But a library of unread books, no matter how well copied, is just a warehouse. The final and most active part of our journey is to understand how this information is used. How does the cell read the blueprint and turn it into a functional, living machine? We are now ready to explore the central dogma, the flowchart that describes the flow of information from storage to action. The central dogma of molecular biology. In our lessons up to this point, we have built a quiet understanding of the blueprint of life. We have explored the physical structure of DNA, the double helix, and we have learned the four-letter language in which its instructions are written. We have just reviewed the incredible process of DNA replication, the molecular machinery that allows this entire vast library to be copied, ensuring that every new cell receives its own perfect set of instructions. We have established that this blueprint is a masterpiece of storage and of continuity. But this raises the most practical question of all. How is this information used? A library full of books, perfectly copied, is of no value if the books are never opened and read. The DNA in your cells contains the recipes for building a human, but how, precisely, is that static, stored information turned into the action of living? How does the blueprint guide the construction of the building? In this lesson, we will explore the fundamental flowchart of life. This concept, first articulated by the brilliant physicist and biologist Francis Crick, is known as the central dogma of molecular biology. It is not a dogma in the sense of a rigid, unproven belief. Rather, it is the foundational statement, the core principle, that describes the flow of genetic information within a biological system. It is the operating system of the cell. The central dogma, in its most essential form, is a simple and profoundly elegant statement. DNA makes RNA, and RNA makes protein. Let that simple phrase settle in your mind. DNA makes RNA, and RNA makes protein. This one-way street of information is the absolute foundation of all function in every living cell. Let us examine each component of this flow. First, we have DNA. As we know, this is the master blueprint. It is the complete, permanent, and priceless instruction manual for the entire organism. In complex cells, which we call eukaryotic cells, this DNA is held precious. It is locked away for its own protection inside a fortified, central compartment called the nucleus. The DNA never leaves this vault. It is far too valuable to be exposed to the chaotic and often harsh chemical environment of the main cell. Its instructions must be preserved, unchanged, for the life of the organism. Second, we have RNA, or ribonucleic acid. Here is the solution to the problem of the locked vault. If the master blueprint cannot leave the nucleus, how does its message ever get to the cell's factories? The cell solves this by making a temporary, disposable copy of one specific instruction. When the cell needs to build one specific thing, it navigates to the DNA blueprint, finds the one recipe or gene it needs, and creates a copy of just that gene. This copy is not made of DNA. It is made of the closely related molecule, RNA. This RNA molecule is a messenger. It is a short, single-stranded, and mobile transcript of the original instruction. Third, we have protein. This is the final product. This is the purpose of the instruction. Proteins are the workers, the machines, and the structural components of your body. When we say DNA codes for your traits, what it truly codes for are proteins. It is the proteins that then create the traits. Proteins are the enzymes that digest your food. They are the hemoglobin that carries oxygen in your blood. They are the keratin that makes up your hair, the collagen that supports your skin, and the antibodies that fight infection. They are the functional things that the DNA blueprint describes. These three players are connected by two great processes. The process of getting from DNA to RNA is called transcription. The word transcribe literally means to write a copy of. This is exactly what the cell does. It transcribes a gene from the permanent DNA language into the temporary RNA language. The second process, getting from RNA to protein, is called translation. The word translate means to change from one language to another. This is also precisely what happens. The RNA messenger carries its code, written in the language of nucleotides, A, U, G, C, to a molecular factory called a ribosome. The ribosome reads this code and translates it into a completely different language, the language of amino acids, which are the building blocks of proteins. So, the central dogma is the full information pathway. The permanent DNA is transcribed into a temporary RNA message. That RNA message then leaves the nucleus, travels to a ribosome, and is translated into a functional protein. To make this flow feel more tangible, let us use a clear analogy. Imagine a vast, ancient, and priceless library. This library represents the nucleus of your cell. In the very center of this library, in a protected, climate-controlled vault, is the master reference collection. This collection is your DNA. It is a set of thousands of rare, massive volumes, your chromosomes, and each volume contains thousands of individual recipes, your genes. The library has one unbreakable rule. The master reference books may never, under any circumstances, leave the protected vault. They are too valuable to be risked in the outside world. Now, imagine you are a builder. You need the recipe for a single specific component, let's say, the recipe for the protein hemoglobin. You cannot take the master book with you. So, you go to the vault. You find the correct volume, the chromosome, and you turn to the correct page, the gene for hemoglobin. The librarian gives you a single piece of scratch paper and a pencil. This piece of paper is RNA. You then carefully, letter for letter, transcribe the recipe from the master book onto your piece of paper. This process is transcription. The master book, DNA, is closed and placed safely back on the shelf, unharmed. Your piece of paper, the RNA, now carries the complete instruction. You take this single piece of paper, this temporary messenger RNA, and you leave the library's vault. You walk out of the nucleus and into the bustling city outside, which represents the main body of the cell, the cytoplasm. You take your transcribed note to a workshop or a factory. This factory is the ribosome. At the workshop, there is a foreman and a team of workers. The foreman, the ribosome, takes your note and reads it. But your note is written in a technical code, the language of nucleotides. The foreman and his workers must translate this code. As they read the instructions, they call for specific parts. We need this part, then this part, then this one. These parts are the amino acids. The workers, other forms of RNA, bring these parts in the correct order, and the foreman, the ribosome, assembles them into a long chain. This chain, once complete, folds up into a functional, three-dimensional machine. You have successfully built your hemoglobin molecule. This is the process of translation. This analogy captures the flow perfectly. The permanent DNA archive is protected. Temporary RNA copies are made of only the needed instructions, and these copies are translated into functional products. This is the fundamental operating system of your cells, running in countless parallel processes right now. This simple flowchart, DNA to RNA to protein, is the core concept that explains how life functions moment to moment. It is how a cell is a cell. It is how your cells respond to their environment. When you are exposed to sunlight, a signal tells your skin cells to turn on the gene for melanin. That gene is transcribed into RNA. That RNA is translated into the protein melanin, which then darkens your skin to protect it. Every thought, every movement, every heartbeat is the result of specific genes being transcribed and translated, creating the specific proteins needed for that exact moment. This dogma is also the key to understanding modern medicine. Many viruses, for instance, are agents that hijack this process. An influenza virus injects its own RNA instructions into your cell, bypassing your DNA entirely. It goes straight to your workshops, your ribosomes, and tricks them into translating the viral message, forcing your own cells to build thousands of new viruses. Conversely, some of the most advanced medicines, such as the mRNA vaccines, are a brilliant and direct application of this dogma. They do not interact with your DNA vault at all. They simply slip a pre-made temporary RNA recipe, the piece of paper from the library, into your cell. This recipe instructs your workshop to build one harmless piece of a virus, just long enough for your immune system to see it, learn to recognize it, and build a powerful defense against it. We have now outlined the grand flowchart of life. We understand that DNA makes RNA, which in turn makes protein. This simple statement, however, contains two massive complex processes, transcription and translation. To truly understand this dogma, we must now zoom in and examine each step. Before we can understand how the message is read, we must first understand the messenger itself. Our next lesson, therefore, will be dedicated to that crucial intermediary molecule, the one that bridges the gap between the protected vault and the busy workshop. We will explore the structure and the different types of ribonucleic acid, or RNA. Ribonucleic acid, RNA, the messenger molecule. Welcome back. In our previous lesson, we established the great flowchart of life, the central dogma. We learned that information flows from the permanent vault of DNA, is copied into a temporary RNA message, and is then read to build a functional protein. This flow, DNA to RNA to protein, is the fundamental operating system of every cell in your body. We have used the analogy of a protected, priceless library, the DNA, and a busy workshop outside the ribosome, but this entire system would fail without a way to bridge the gap. We need a messenger, a runner, a copyist who can take the precious instructions from the vault and deliver them to the factory floor. This messenger is ribonucleic acid, or RNA. It is the crucial intermediary, the pivot point upon which the entire central dogma turns. To understand how life functions, we must first understand this versatile and essential molecule. This lesson is dedicated entirely to RNA, its unique structure, and its many forms. Let us begin by defining our terms. What is ribonucleic acid? Like DNA, it is a nucleic acid, a polymer, a long chain of nucleotides. It looks, at first glance, very similar to its more famous cousin DNA, but if we look closer, we find three small, yet profound, chemical differences. These differences are not accidental. Each one perfectly adapts RNA for its role as a temporary, active, and mobile message, just as DNA's structure makes it perfect for permanent, stable storage. The first difference is in the sugar. The D in DNA stands for deoxyribose. The R in RNA stands for ribose. This is the sugar that forms the backbone of the molecule. The only difference between them is that ribose has one extra oxygen-hydrogen group. The sugar in DNA is deoxy, meaning it is missing that oxygen. This single atom makes RNA more chemically reactive, less stable, and more easily broken down by the cell. For a permanent blueprint, this would be a disaster. But for a temporary message that needs to be deleted after it is read, this instability is a feature, not a bug. The second key difference is its structure. DNA, as we know, is the double helix. It is a stable, twisted ladder, with its two strands locked together. RNA, by contrast, is typically single-stranded. It is like a single side of the ladder, just one long chain, one sugar phosphate backbone with its bases, A, U, G, C, hanging off the side. This single-stranded nature is the key to RNA's versatility. It is not a rigid rod, it is a flexible ribbon. The third difference is in its alphabet. RNA uses almost the same four-letter code as DNA, but with one substitution. It has adenine A, guanine A, G, and cytosine C, but it does not have thymine A, T. Instead, it uses a very similar base called uracil, U. Uracil is, in effect, a stand-in for thymine. It serves the exact same purpose. It forms base pairs with adenine, A. So, in the world of RNA, the base pairing rule is not AT, but AU. G, as always, still pairs with C. These three changes, the ribose sugar, the single-stranded form, and the use of uracil, define the RNA molecule. They make it a perfect, transient copyist for the DNA's permanent message. That single-stranded, flexible nature of RNA is where its true magic lies. Unlike the rigid DNA ladder, an RNA ribbon can fold. It can fold back on itself. Parts of the same RNA strand can form base pairs with other parts of the same strand. An A, in one part of the chain, can find a U 50 bases later and form a weak bond. A G can find a C. This folding allows an RNA molecule to twist and contort itself into complex, stable, three-dimensional shapes. It can form hairpin loops, cloverleafs, and intricate, globular structures. This is a profound concept, because in biology, shape is function. By folding into specific shapes, RNA can do things. It is not just a passive message, it can be an active machine. This functional diversity gives us not one, but several main types of RNA in the cell, all working together to achieve the central dogma. We will focus on the three most important players. First, there is messenger RNA, mRNA. This is the molecule that truly lives up to the name messenger. This is the direct copy, the transcript, of a single gene from the DNA. In our library analogy, this is the piece of paper with the recipe. It is a long, straightish ribbon whose sole purpose is to carry the codon sequence from the nucleus to the ribosome. It is the message in messenger RNA. Second, there is transfer RNA, tRNA. This is the translator. It is the physical bridge between the two languages. A tRNA molecule is a small, specific piece of RNA that folds into a precise cloverleaf shape. At one end of this shape, it has a three-letter sequence called an anticodon. This anticodon is complementary to a codon on the mRNA. For example, if the mRNA codon is AG, the tRNA that binds to it will have the anticodon UC. At the other end of the tRNA molecule is a binding site for one, and only one, specific amino acid. In this case, the UUC chip tRNA will always carry the amino acid lysine. The tRNA, therefore, is the physical adapter that reads the nucleotide word and transfers the correct amino acid part. Third, there is ribosomal RNA, rRNA. This is the most abundant type of RNA in the cell, and perhaps the most surprising. Ribosomal RNA, along with a few proteins, forms the physical structure of the ribosome itself. The ribosome, our factory, is not just made of protein. It is, in fact, mostly made of RNA. The rRNA molecules are not just a passive scaffold, they are the active catalytic core. It is the rRNA that orients the mRNA message and the tRNA translators. And it is the rRNA that acts as an enzyme, a ribozyme, that physically forms the new chemical bond that links one amino acid to the next, building the protein chain. The factory itself is an RNA machine. This discovery that RNA can be an enzyme was a Nobel Prize-winning breakthrough. It completely changed our understanding of life. For decades, we believed in a strict division of labor. DNA stores information, and proteins do the work, as enzymes. But RNA, we now know, can do both. This leads to a beautiful and powerful theory known as the RNA world hypothesis. This is the idea that billions of years ago, before DNA and before proteins, life may have begun with RNA. It was an RNA world. A single RNA molecule could have both stored genetic information in its A, U, G, C sequence and catalyzed its own replication by acting as a ribozyme. It was a single molecule solution to life. Over eons, evolution offloaded these jobs. The more stable DNA took over the storage role, and the more efficient proteins took over the catalytic role, leaving RNA as the essential intermediary we see today. This ancient, versatile nature of RNA is still visible everywhere. We see it in modern mRNA vaccines, which we've discussed. These vaccines are a direct application of the central dogma. We are not injecting a virus, we are simply giving our cells a pre-made mRNA recipe. Our own ribosomes, RNA, and our own tRNAs translate this message, build a harmless practice protein, and in doing so, train our immune system for the real thing. We also see RNA's power in many viruses. Viruses like influenza, HIV, and the common cold do not even use DNA. Their entire genetic code is stored as RNA. They are RNA viruses. They hijack our cellular machinery, using our ribosomes to directly translate their RNA into new viral proteins, bypassing our DNA vault entirely. We have now assembled our team. We understand the blueprint, DNA, and we have been introduced to the three key players of its execution: the messenger, mRNA, the translator, tRNA, and the factory, rRNA. We know who the messenger is. The next logical step in our journey is to understand how that messenger is created. How does the cell find the one correct gene among billions of letters in the DNA vault and transcribe it into that single, perfect mRNA copy? This process, the first great step of the central dogma, is called transcription. Our next lesson will explore how we get from DNA to RNA. Transcription, from DNA to RNA. Welcome back. Our journey has brought us to the very heart of the central dogma, the moment of action. We have our complete, protected library of instructions, the DNA, and we have met the versatile and essential messenger molecule, RNA. We know what the blueprint is, and we know who the messenger is. Now, we must ask the critical question: how is that message created? How does the vast, complex machinery of the cell navigate the billions of letters in the DNA vault, find the one correct recipe or gene that it needs at this exact moment, and then write a copy of it into a new RNA molecule? This process, the first great step in the flow of information from storage to function, is called transcription. The word itself, to transcribe, means to copy something from one form to another. In this lesson, we will explore, step by step, how the cell's molecular machinery reads the permanent DNA code and transcribes it into a temporary, mobile mRNA message. The entire process of transcription is managed by a single, large, and magnificent enzyme called RNA polymerase. This protein is the master copyist of the cell. It is the monk in our library analogy, the one who will carefully create the scroll from the master book. The process, like many in biology, can be understood in three distinct phases: initiation, elongation, and termination. First, initiation, or the beginning. The RNA polymerase enzyme cannot simply land anywhere on the DNA and start copying. It must be told where to begin. On the DNA strand, just upstream, or before the start of every gene, there is a special landing strip, a sequence of bases known as the promoter, This promoter region is a signpost. It essentially broadcasts a signal that says, a gene starts here. The RNA polymerase, along with other helper proteins, recognizes and binds tightly to this promoter sequence. Once securely attached, the RNA polymerase begins the second phase, elongation, or the building of the message. The enzyme performs several tasks at once. First, it unwinds a small local section of the DNA double helix, prying the two strands apart. This creates a small, temporary transcription bubble. This bubble exposes the sequence of bases on the two strands, making them readable. Here, a critical choice is made. RNA polymerase only reads one of the two DNA strands. This strand is called the template strand. The other strand, the one not being read, is called the non-template or coding strand. The RNA polymerase reads this template strand one base at a time. As it moves, it grabs free-floating RNA nucleotides A, U, G, and C from its environment. Following the rules of complementary base pairing, it matches them to the DNA template. If it reads an A on the DNA, it adds a U, uracil, to the new RNA strand. If it reads a G, it adds a C. If it reads a C, it adds a G. And if it reads a T on the DNA, it adds an A. In this way, the enzyme builds a new, single strand of RNA that is a perfect, complementary copy of the DNA template. This new strand, the messenger RNA, mRNA, grows longer, one nucleotide at a time, as the RNA polymerase moves down the gene. Behind the enzyme, the DNA double helix zips itself back up, perfectly unharmed, and the new RNA ribbon trails off behind it. Finally, the third phase, termination, or the end. The RNA polymerase cannot copy forever. Just as there is a start signal, there is a stop signal. At the very end of the gene, there is another special sequence of bases on the DNA, called the terminator. When the polymerase reaches this sequence, it is a signal that the recipe is complete. The enzyme detaches from the DNA, and the newly synthesized mRNA molecule is released. The transcription bubble closes, the DNA is fully rewound, and the master book is now back on the shelf, exactly as it was. We are left with one new, complete mRNA scroll. This core process is elegant, but in complex cells like our own eukaryotic cells, this is only the beginning. The mRNA molecule that is first created by RNA polymerase is not a finished product. It is a rough draft, a pre-mRNA. It must go through a vital editing and maturation process before it is allowed to leave the nucleus. This editing is called RNA processing. This necessity arises from a strange and fascinating feature of our genes. Most of our genes are not continuous, uninterrupted recipes. Instead, they are fragmented. They contain long, non-coding, junk sequences, interrupting the actual, useful instructions. The useful coding parts are called exons because they are expressed. The interrupting, junk parts are called introns because they are intervening sequences. The first step of processing, therefore, is splicing. The cell must cut out all the useless introns and stitch the useful exons together in the correct order to create one continuous, coherent message. This is an incredibly precise molecular surgery. It is performed by a complex machine called the spliceosome, which is itself made of small RNA molecules and proteins. The spliceosome recognizes the boundaries between exons and introns, loops the intron out, cuts it, and splices the two exons together, leaving a seamless, functional recipe. After splicing, the cell adds two more features to the mRNA to protect it for its dangerous journey outside the nucleus. At the front of the message, the 5' end, it adds a 5 cap. This is a special, modified guanine nucleotide that acts like a passport. It will help the ribosome recognize and dock with the mRNA once it reaches the cytoplasm. At the back of the message, the 3' end, the cell adds a 3' A tail. This is a long, a long chain of hundreds of adenine nucleotides. This tail acts as a buffer or a timer. In the cytoplasm, enzymes are waiting to chew up and degrade RNA. This tail is a decoy. The enzymes will chew through the long tail first, giving the important, coded message in the middle enough time to be read and translated by a ribosome, perhaps several times, before it is finally destroyed. Only after this splicing, capping, and tailing is the molecule considered a mature mRNA. It is now, finally, ready. It is given its exit visa and is actively transported out of the nucleus through a small gateway called a nuclear pore and into the bustling cytoplasm in search of a ribosome. This entire process of transcription is the master control point for all of life. Think back to our earlier question. Why is a brain cell different from a skin cell if both have the same DNA? The answer is differential transcription. A brain cell is a brain cell because it turns on or transcribes the genes for neurotransmitters while keeping the genes for skin pigment turned off. A skin cell does the opposite. This on-off switch is often a set of helper proteins called transcription factors. These proteins bind to the DNA near the promoter and either help or block the RNA polymerase from landing. Every signal your body receives a hormone, a ray of sunlight, the food you eat can trigger these transcription factors, thus changing which genes are being transcribed at any given moment. This is how your cells adapt. This process is also a critical target for medicine. Many of our most effective antibiotics work by specifically attacking this process. For example, the antibiotic rifampicin works by binding to and jamming the bacterial RNA polymerase. It has no effect on our human RNA polymerase, but it completely stops the bacterium from transcribing its genes, thus preventing it from making the proteins it needs to survive. We have now completed the first great step of the central dogma. We have seen how the RNA polymerase finds the correct gene, how it transcribes the DNA into a pre-mRNA, and how that rough draft is meticulously processed, spliced, capped, and tailed into a mature, finished message. This message is now complete. It has left the safety of the nucleus and is in the cytoplasm. It carries the complete coded recipe for a protein. All it needs is a workshop and a translator. Our next lesson explains what happens when this mRNA molecule finds a ribosome. We will explore the final, beautiful process of translation. Translation from RNA to protein. In our last lesson, we completed a great journey. We followed the creation of a mature messenger RNA, mRNA, molecule as it was transcribed from the DNA blueprint, meticulously processed, spliced, capped, and tailed. We left it as it passed through the nuclear pore, exiting the protected library of the nucleus and entering the bustling, active city of the cytoplasm. This message, this scroll with the copied recipe, is now floating in the cellular soup. It carries the complete, coded instructions for building a protein. But the message itself is useless without a workshop to read it and a translator who understands its language. This is the moment where the blueprint becomes a building. This is where the code becomes life. In this lesson, we explore the second and final great step of the central dogma. We will learn how the cell translates the language of nucleotides written on the mRNA into the completely different language of amino acids, the building blocks of a protein. This beautiful and complex process is called translation. Let us begin by defining the act of translation. It is the synthesis of a protein from an mRNA template. It is the conversion of information from one chemical language to another. This entire monumental task is performed by a set of remarkable molecular machines. The first and most central player is the ribosome. This is the workshop or factory we have spoken of. The ribosome is a large, complex structure made of its own ribosomal RNA, rRNA, and proteins. It is not a passive workbench. It is an active, grasping machine. It is designed to grab onto the mRNA message, hold it steady, and read its code, one codon at a time. The second player is the mRNA itself, the message. It is the instruction tape, the blueprint, that will be fed through the ribosome machine. Its language, as we know, is written in three-letter words called codons. The third, and perhaps most clever, player is the transfer RNA, tRNA. This is the translator, the adapter, the bilingual specialist. As we learned, each tRNA molecule has two critical ends. At one end, it has a three-letter sequence called an anticodon, which is the perfect complement to a codon on the mRNA. For example, if the mRNA codon reads AUG, the tRNA that recognizes it will have the anticodon UAC at its other end. This specific tRNA is chemically bound to the one amino acid that the AUG codon codes for, in this case, methionine. There is a whole fleet of these tRNAs in the cell, one for each possible codon, each carrying its one correct amino acid. The process of translation, then, is a dance between these three players. The ribosome grabs the mRNA and reads the first codon. The correct tRNA, the one with the matching anticodon, arrives and docks with that codon, bringing its specific amino acid with it. The ribosome then moves to the next codon. The next correct tRNA arrives, bringing its amino acid. The ribosome, acting as an enzyme, takes the first amino acid and chemically links it to the second one, forming the beginning of a chain. The first tRNA, now empty, is released. The ribosome moves to the third codon. The third tRNA arrives. The ribosome links the growing chain onto this third amino acid. This cycle repeats, over and over. Codon by codon, the mRNA is read. tRNA by tRNA, the correct amino acids are delivered. And link by link, the ribosome stitches them together into a long chain called a polypeptide. This chain, once it is complete, will fold up into a functional protein. Let us explore this molecular dance in greater detail. Like transcription, translation proceeds in three distinct phases: initiation, elongation, and termination. The process begins with initiation. The ribosome is actually made of two separate pieces, a small subunit and a large subunit, which float separately in the cytoplasm. First, the small ribosomal subunit, along with a special initiator, tRNA, the one carrying methionine, which corresponds to the start codon, AU, binds to the front of the mature mRNA. It scans along the message until it finds that specific start codon, AU. This codon is the universal signal for begin translation here. Once it finds the AUG, the large ribosomal subunit clamps down on top, sandwiching the mRNA and the initiator tRNA. The entire, complete factory is now assembled and ready to work. Now, the main phase begins, elongation. This is the cycle of building the chain. To understand this, you must picture the large ribosomal subunit as having three slots or dockets inside it. These are known as the A site, the P site, and the E site. At the start, our initiator tRNA is sitting in the middle slot, the P site, which stands for polypeptide. The first codon, AUG, is in this site. The second codon on the mRNA is now exposed in the first slot, the A site, which stands for aminoacyl, or arrival. The correct tRNA for that second codon flies in from the cytoplasm and docks in the A site. Now, we have two tRNAs side by side. The P site holds the tRNA with the first amino acid. The A site holds the tRNA with the second amino acid. The ribosome itself then performs its most magical trick. The rRNA inside it acts as an enzyme, breaking the bond between the first amino acid and its tRNA in the P site, and simultaneously forming a new peptide bond between that first amino acid and the second one in the A site. The growing polypeptide chain has now been transferred to the tRNA in the A site. The ribosome then performs a physical action called translocation. It moves one codon length down the mRNA. This single clunk shuffles everything over. The now empty tRNA that was in the P site is moved to the last slot, the E site, for exit, from which it is ejected back into the cytoplasm to be recharged with a new amino acid. The tRNA that was in the A site, now holding the growing 2 amino acid chain, is moved into the P site, and the A site is now empty again, exposing the third codon of the mRNA. The cycle begins again. A new correct tRNA arrives at the open A site. The ribosome links the growing chain, now 2 amino acids long, from the P site onto the new amino acid in the A site. The ribosome clunks over one more spot. The empty tRNA exits from the E site. The P site is filled. The A site is open. This process continues, fast and repetitive, adding 1 amino acid every fraction of a second, building a long polypeptide chain that snakes out of a tunnel in the ribosome. Finally, we have termination. This elongation cycle cannot continue forever. The mRNA recipe has an end. This is marked by one of 3 special stop codons, UAA, UAG, or UGA. These codons do not code for any amino acid. There is no tRNA in the cell with an anticodon to match them. Instead, when a stop codon slides into the A site, a protein called a release factor binds there. This release factor acts like a wrench in the gears. It causes the ribosome to stall and triggers the entire assembly to fall apart. The ribosome's enzymatic machinery cuts the completed polypeptide chain free from the last tRNA. The chain, now a finished protein, floats away to do its job. The ribosome subunits separate from each other and from the mRNA. The mRNA itself is now free, and it will likely be grabbed by another ribosome to start the whole process over again, at least for a while, before it is eventually degraded. This process, translation, is the literal creation step of life. It is the physical assembly line that builds you. Every protein that forms your body and makes it function, the hemoglobin carrying oxygen in your blood, the enzymes digesting your food, the collagen in your skin, the keratin in your hair, the antibodies fighting off this morning's cold virus. Every single one of those molecules was built, amino acid by a ribosome reading an mRNA message. Because this process is so fundamental and so different in bacteria versus humans, it is one of the single most effective targets for medicine. Many of our most powerful antibiotics work by attacking the bacterial ribosome, while leaving our own human ribosomes unharmed. For example, tetracycline blocks the A site, preventing new tRNAs from even arriving. Erythromycin physically jams the ribosome, preventing it from translocating. They are a perfect, targeted sabotage of the bacterial factory, shutting down its protein production and killing the infection. We have now followed the flow of information to its ultimate conclusion. We have gone from the DNA master book to the RNA scroll, and from that scroll to the finished product, the protein. We have discussed the entire process, but we have been using one word rather loosely, the word gene. We have called a gene a recipe, or a unit of information. But what is a gene in the precise molecular terms we have just learned? Is it just the part that is translated? Or does it include the promoter? What about the introns that get cut out? Our next lesson will provide a clear, modern definition for this fundamental unit of information. What is a gene? Defining the unit of information. Welcome back. Throughout our journey, we have followed the flow of information from its storage to its ultimate expression. We have seen the DNA blueprint, the RNA messenger, and the protein product. And all along we have used a single convenient word to describe the recipe at the heart of this process: the gene. We have used this word in an intuitive sense, calling it a unit of information or the instructions for a trait. But now that we understand the intricate mechanical processes of transcription, RNA processing, and translation, we must return to this simple word and ask a more difficult, more precise question. What, exactly, is a gene? Is it just the part of the DNA that is read by the ribosome? Does it include the junk DNA that gets spliced out? What about the start and stop signals? The simple classical definition of a gene is no longer sufficient. In this lesson, we will move beyond the analogy and build a clear, modern, and molecular definition that incorporates all the complex machinery we have just learned. We can begin with the classical definition, the one understood by Gregor Mendel as he studied his pea plants. In this view, a gene is the fundamental, abstract unit of heredity. It is a factor that is passed from parent to offspring and controls a single, observable trait, like the color of a flower or the texture of a seed. This definition, while historically important, is insufficient for a molecular biologist. The trait of purple flower color, for example, is a very high-level outcome. It is the end result of a complex chemical pathway run by multiple enzymes, which are themselves built by multiple recipes. The classical definition describes the effect, but not the object. To find the object, we must look at the functional product. The central dogma showed us that the recipe in the DNA is used to build a product. We saw this in our library analogy. The recipe in the master book is transcribed and translated to build a final machine or product. So, a modern molecular definition begins here. A gene is a specific sequence of nucleotides in DNA that provides the instructions for making a specific functional product. For many decades, it was assumed this functional product was always a protein, and in most cases, this is true. A gene's DNA sequence is transcribed into an mRNA, and that mRNA is translated into a polypeptide chain, which then folds into a functional protein. This protein then does something. It acts as an enzyme, or a structural component, or a signal, and in doing so, contributes to a trait. But this is not the whole story. As we learned when we met the actors in our translation story, the cell also makes functional products that are not proteins. Think back to the transfer RNA, tRNA molecule. This is the translator that brings the correct amino acid to the ribosome. The cell must make this tRNA. It does so by reading a gene for tRNA on the DNA. This gene is transcribed into an RNA molecule, but this RNA molecule is never translated. Its job is not to carry a code. Its job is to be a machine. It folds into its functional cloverleaf shape and goes to work. Its function is to be an RNA. The same is true for ribosomal RNA, rRNA. The gene for rRNA is transcribed from the DNA, and the resulting RNA molecule is used as a structural and enzymatic component of the ribosome itself. It, too, is never translated into a protein. Therefore, our molecular definition of a gene must be broader. The functional product is not always a protein. The complete, modern definition is this. A gene is a defined region of DNA that produces either a polypeptide, which folds into a protein, or a functional RNA molecule. This definition is good, but we can make it even more precise. What parts of the DNA are included in this defined region? Let us return to our cookbook analogy. If a gene is a recipe, is the recipe just the ingredient list and the step-by-step instructions? Or, to be useful, must the recipe also include its title, its introduction, and the bake at 350 degrees instruction at the top? A functional recipe needs all of these things. A functional gene does too. Let's consider the promoter. This is the landing strip or start signal on the DNA before the transcribed part. The promoter sequence itself is not copied into the RNA message. But without it, the RNA polymerase enzyme would never know where to begin. The gene would be silent and useless. Therefore, most modern biologists consider the promoter region to be an essential part of the gene's address or title page. It is a regulatory region, and it is part of the gene. Now, what about the introns? These are the long intervening sequences within the gene that are transcribed into the rough draft pre-mRNA, but are then meticulously spliced out and discarded. They never make it into the final, translated message. Are they part of the gene? Yes. The gene, as a unit on the DNA, is the entire sequence from start to finish that is transcribed. The introns are a part of that initial transcribed unit. The gene on the DNA is the full, fragmented, unedited version of the recipe. Finally, we have the terminator sequence. This stop signal at the end of the gene tells the RNA polymerase to fall off. Like the promoter, it is not part of the final protein product, but it is an essential instruction that defines the end of the gene. So, a truly complete structural definition of a gene is this. A gene is the entire DNA sequence required to produce a single functional product. This includes not only the coding sequences that are translated, the exons, but also the non-coding sequences that are transcribed, the introns, and the regulatory sequences that control its transcription, such as the promoter and terminator. It is important to know that this definition is still a subject of scientific refinement. Some definitions are more narrow, counting only the transcribed region. Other, even broader, definitions include enhancer sequences, which are other pieces of DNA that can be thousands of letters upstream or downstream, but still act as switches to help turn the gene on or off. The concept of a gene, it turns out, is wonderfully fuzzy at the edges, blending into the regions around it. This precise modern definition is not just an academic exercise, it has profound, real-world consequences. When we talk about the gene for a specific trait, like cystic fibrosis, what we really mean is the gene promoter, exons, and introns that codes for the CFTR protein, which is a channel that transports chloride ions across cell membranes. Understanding this complexity is vital for diagnosing disease. A person can have a genetic disease without having a typo in the recipe, the exons, at all. For example, some forms of cystic fibrosis are caused by a mutation in an intron. The exons are perfect, but the mutation breaks the splices signal. This causes the cell's splicing machine, the spliceosome, to make a mistake. It might accidentally cut out an exon along with the introns, or leave an intron in. The resulting mRNA is garbled, and the translated protein is broken and non-functional. The disease is caused by an error in a non-coding region. Similarly, a mutation in the promoter region can cause disease. The exons, the recipe might be perfect, but if the title page is illegible, the RNA polymerase cannot bind. The gene is never turned on, the protein is never made. The person has a disease of gene regulation, not of protein sequence. This is also why genetic engineering is so complex. When scientists want to insert a human gene, like the one for insulin, into a bacterium, they cannot just move the exons. They must insert a complete gene cassette, a piece of DNA containing a promoter, one that a bacterium can read, the coding sequence, and a terminator. They are building a complete, functional unit of information. We have now arrived at a complete, modern, and wonderfully complex definition of the gene. It is not just a recipe, but a complete, self-contained functional unit with a title, regulatory switches, edited instructions, and a stop signal. We have used the analogy of a giant cookbook, the genome, which contains thousands of these individual recipes, or genes. But a good chef does not use every recipe in the cookbook at the same time. You do not bake a cake, roast a turkey, and boil pasta all at once. You select the recipes you need at the time you need them. Cells are the same. Every cell in your body has the same, complete cookbook. But a skin cell is a skin cell because it only uses the recipes for skin proteins. A muscle cell is a muscle cell because it only uses the recipes for muscle proteins. This leads us to the next, and most dynamic, question of all. How does a cell control which genes are on and which are off? This is the topic of our next lesson: gene expression and regulation. Gene expression and regulation. Welcome back. In our last lesson, we arrived at a clear molecular definition of a gene. We understand it as a complete functional unit on the DNA, a recipe that includes not just the instructions, but also the start and stop signals and all the necessary components to produce a final, functional product. This understanding, however, leads us to one of the deepest and most beautiful paradoxes in all of biology. Every single cell in your body, with very few exceptions, contains the exact same complete set of genes. The DNA in your brain cells is identical to the DNA in your skin cells, which is identical to the DNA in your muscle cells. They all possess the same, complete cookbook. And yet, a neuron is profoundly different from a skin cell. They look different, they have different jobs, and they build different materials. How can this be? If every cell has the same library, how do they know what to become? The answer is that having the recipe is not the same as using it. A library may contain books on every subject, but you only check out and read the ones you need. This selective reading, this cellular choice, is the topic of our lesson. We will explore the dynamic and elegant world of gene expression and regulation. Let us first define our two key terms. Gene expression is the process of a gene being used. It is the entire workflow we have studied, from transcription, copying the mRNA, to translation, building the protein. When a gene is on, we say it is being expressed. That expression can be high, making a lot of product, or low, making just a little. Gene regulation, then, is the control of this process. It is the vast and intricate system of switches, dials, and signals that determines which genes are expressed, when they are expressed, and how much they are expressed. To return to our cookbook analogy, every cell has the complete 10,000-page cookbook. Gene regulation is the chef, the cell, deciding that tonight we will only use the recipes from Chapter 4, the skin recipe, and we will ignore all the recipes from Chapter 1, 2, the brain recipes. A skin cell, therefore, is a skin cell not because it lacks the genes for a neuron, but because it is actively expressing the genes for skin proteins like keratin, while actively silencing or repressing the genes for neural proteins like neurotransmitters. A brain cell does the exact opposite. This differential gene expression is the root of all cellular identity. So, how does a cell physically flip a switch on a gene? There are many control points the cell can block translation or destroy an mRNA after it's made, but the most important and most efficient control point is the very first step: transcription. The cell's logic is simple. Why waste energy building an mRNA copy if you do not need the protein? The most effective way to keep a gene off is to prevent the RNA polymerase, our copyist enzyme, from ever binding to the gene's promoter, its start signal. This is achieved by proteins called transcription factors. These are the hands that flip the switches. They can work in two main ways. First, there are repressors. A repressor is a transcription factor that turns a gene off. It works by binding to a special sequence of DNA near the promoter, called an operator. When the repressor is attached, it physically blocks the RNA polymerase. It is a gate in the down position. The RNA polymerase can see the gene, but it cannot land and begin transcription. The classic example is the lac operon in the bacterium E. coli. An operon is a set of genes that are all controlled by one switch. The lac operon contains the genes for enzymes that digest lactose, or milk sugar. When there is no lactose present, the bacterium does not want to waste energy making these enzymes. So, a repressor protein is active. It binds to the lac operon's operator and physically blocks transcription. The genes are off. But when you drink milk and lactose is present, the lactose molecule itself, or a byproduct, acts as a key. It binds to the repressor protein, causing the repressor to change its shape and fall off the DNA. The gate is now up. The RNA polymerase can rush in, bind to the promoter, and transcribe the genes. The genes are on. The bacterium produces the enzymes it needs to eat the lactose. This is a perfect, simple example of gene regulation in response to the environment. Second, there are activators. These are transcription factors that turn a gene on or amplify its expression. They bind to the DNA and act like a beacon, helping the RNA polymerase to bind to the promoter. They stabilize the enzyme and make transcription far more efficient. In our complex human cells, this process is layered with even more sophistication. This is epigenetics, which means above or on top of genetics. Epigenetics refers to changes in gene expression that do not involve changing the DNA sequence itself. It is all about access. Your DNA is not a loose floating strand. It is a vast thread, 2 meters long, packed into a microscopic nucleus. To do this, it is tightly wound around spools of protein called histones. This DNA histone complex is called chromatin. For a gene to be read, it must first be physically unspooled. This packing itself is a form of gene regulation. When the DNA is wound tightly around the histones called heterochromatin, the RNA polymerase cannot even get to the gene. The entire chapter of the cookbook is packed away in a locked box. This is a long-term off switch. When the DNA is wound, loosely called uchromatin, the promoter and gene are exposed, accessible, and ready to be read. This is the on state. The cell controls this tightness by adding small chemical tags to the histones, like labels on the spool. A process called acetylation, adding an acetyl group, typically loosens the coil, turning genes on. Conversely, the cell can add a chemical tag, a methyl group, directly onto the DNA itself. This DNA methylation is a powerful off switch. It recruits proteins that lock the chromatin down, making it dense and unreadable. This is how a skin cell ensures its brain genes are not just off, but permanently packed away for the life of the cell. This dynamic, multi-layered system of gene regulation is not an abstract concept. It is the story of your own life. Your development from a single fertilized egg into a complex being of trillions of cells was, and is, a precisely choreographed dance of gene regulation. Specific genes were turned on in sequence, telling cells, you will become a heart, you will become a nerve, you will become a bone. This system is also the key to your moment-to-moment adaptation. When you exercise, you send signals to your muscle cells. These signals activate transcription factors that turn on the genes for muscle growth and repair. When you are stressed, the hormone cortisol activates transcription factors in your cells to change their metabolic state. And, tragically, this system is the root of many diseases. Cancer is, at its heart, a disease of broken gene regulation. It is a disease where the on-off switches have failed. Genes that tell the cell, divide, 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 called proto-oncogenes, can get a mutation that locks them in the on position. Conversely, genes that say stop dividing, check for errors, called tumor suppressor genes, can get a mutation that locks them in the off-F position. This combination of stuck accelerators and broken brakes is what causes the uncontrolled cell growth that defines cancer. We have now seen how the cell, in its wisdom, controls its vast library. We understand it does not just have genes, it manages them, turning them on and off with transcription factors and locking them away with epigenetic tags. We have even touched upon how the DNA is packed, tightly wound around histone spools. This brings us to a final, profound question of sheer scale and organization. We have been talking about genes and promoters, but these are just tiny sentences and paragraphs in a library of billions of letters. How does a cell organize this vast, 2-meter-long thread of DNA? How is this cookbook physically bound, sorted, and duplicated? We must now zoom out from the single gene and look at the entire massive packaging of DNA. This is the structure of the chromosome. Chromosomes, the packaging of DNA. Welcome back. In our recent lessons, we have been zoomed in, operating at the level of the single gene. We have learned how a gene is defined, how it is regulated, and how it is turned on and off. But now, we must zoom out all the way out to confront a profound physical problem. The human genome, the complete set of instructions, is a DNA molecule of roughly 3 billion base pairs. If you were to take the DNA from a single one of your microscopic cells, untangle it, and stretch it out into one long continuous thread, it would be about 2 meters long, over 6 feet. This 2-meter long, impossibly thin thread must be packed, organized, and stored inside the nucleus, a cellular compartment so small it is almost invisible to the naked eye. This is a feat of data compression that is, by any scale, a miracle of engineering. It is like packing 40 kilometers or 25 miles of the finest thread into a single tennis ball. How is this possible? And how can the cell perform this incredible packing feat while still keeping its recipes, the genes, accessible for reading? This lesson explores the remarkable, multi-layered packaging system of DNA. We will explore the chromosome. DNA is not, as one might imagine, a loose tangle inside the nucleus, like a bowl of spaghetti. It is an active, dynamic, and highly organized structure. The entire system is built upon a partnership between the DNA and a set of proteins, the most important of which are called histones. The histones are the spools. They are small, positively charged proteins. DNA, with its phosphate backbone, is a strongly negatively charged molecule. This creates a powerful natural attraction. The packaging process begins at the first and most basic level. The long, 2-meter strand of the DNA double helix is wrapped, like thread, around a set of these histone spools. The basic, repeating unit of this packaging is called a nucleosome. Picture a bead made of 8 histone proteins. The DNA strand wraps around this bead about 1 2/3 times, then continues on to the next bead, and the next, and the next. The resulting structure looks like beads on a string. This first step alone condenses the DNA's length about 7-fold. But this is only the beginning. The beads on a string are then coiled up, like a spring. This string of nucleosomes is wound into a thicker, denser fiber, about 30 nanometers in diameter, known as a solenoid. This coil of coils condenses the DNA even further. This 30 nanometer fiber is still not compact enough. It is then organized into a series of vast loops, which are anchored onto a central protein scaffold. Imagine this thick, coiled rope being looped back and forth, like a long extension cord being neatly gathered. This entire DNA and protein complex, the string, the spools, the coils, and the loops is known collectively as chromatin. This is the normal state of your DNA inside the nucleus of a cell that is simply living, growing, and doing its job. This chromatin is not uniform. As we touched upon in our last lesson, it exists in 2 forms. The loops can be uncoiled and opened up, creating a loose, fluffy state called euchromatin or true chromatin. In this state, the DNA is accessible. The promoters and genes can be reached by RNA polymerase. This is the active chromatin, where genes are on. Alternatively, the loops can be packed down tightly upon themselves, creating a dense, compact state called heterochromatin. This is the inactive, locked, or silent chromatin. The genes in this region are inaccessible, packed away in long-term storage. This chromatin, a dynamic structure of loops and coils, is how your DNA exists for almost all of its life. However, there is one final, more extreme level of condensation. This happens only when a cell is preparing to do one specific, critical thing: divide. When a cell prepares to divide, a process called mitosis, it must first copy its entire genome. It performs DNA replication. It now has 2 full, identical copies of its 2-meter-long DNA. To ensure that each new daughter cell gets one perfect, complete copy, the cell must sort this information flawlessly. Trying to sort 2 6-foot-long tangles of chromatin would be an impossible, catastrophic mess. So, the cell performs a final, miraculous act of packing. It takes the looped chromatin fibers and coils them, and coils them, and coils them again, condensing them into the densest possible, most transportable form. This final, fully condensed X-shaped structure is what we traditionally recognize as a chromosome. It is a critical and common point of confusion. The X shape we see in textbooks is not the normal state of DNA. It is a temporary, duplicated, shipping container state, visible only for the brief window of cell division. This X shape, more properly called a metaphase chromosome, is made of two identical copies. Each side of the X is one of the copies. We call them sister chromatids. They are physically joined at a waist region called the centromere. This X shape is a temporary package designed for sorting. During division, this structure is grabbed at the centromere by the cell's machinery. The X is split down the middle, and the two identical sister chromatids are pulled apart, one to each new cell. Each new cell, therefore, gets one complete, unduplicated copy, which then unspools back into its functional, fluffy chromatin state. This packaging system is the basis of your genetic legacy. The human cookbook is not one single 2-meter-long strand. It is broken up into manageable volumes. Each volume is one chromosome. In a normal human body cell, a somatic cell, you have 46 of these volumes. They are organized into 23 pairs. You received one complete set of 23 chromosomes from your mother in the egg, and one complete set of 23 from your father in the sperm. These matching pairs are called homologous chromosomes. They are homologous because they are the same size, the same shape, and they carry the same genes in the same order, though they may have different versions, or alleles, of those genes. Scientists can take a snapshot of these 46 chromosomes from a dividing cell and arrange them by size, from largest, chromosome 1, to smallest, chromosome 22. This organized family portrait of your chromosomes is called a karyotype. These first 22 pairs are called autosomes. The 23rd pair are the sex chromosomes. In females, this pair consists of two X chromosomes, XX. In males, it consists of one X and one, much smaller, Y chromosome, XY. The importance of this perfect packaging and sorting system is made tragically clear when it fails. Sometimes, during the creation of an egg or sperm, an error occurs, and the chromosomes are not sorted correctly. A new individual might be born with a missing chromosome, or an extra one. This condition is called aneuploidy. A common example is Down syndrome, which is caused by the presence of a third copy of chromosome 2, one also called trisomy 21. This one extra volume in the library disrupts the entire balance of gene expression, leading to the characteristic effects of the condition. We have now seen how the genome is organized, from the simple base pair to the gene to the entire, massive, coiled structure of the chromosome. We have seen how this structure is essential for life, for regulation, and for the orderly division of cells. But this system, from replication to sorting, is a physical, chemical process. It is incredibly accurate, but it is not perfect. Errors can happen. The DNA itself, this precious molecule, can be damaged by its environment. Our final lesson will explore what happens when the code itself, the sequence of letters in the book, is changed. We will now discuss mutations. Mutations, changes in the genetic code. In our final lesson on this topic, we arrive at an inevitable reality of any complex information system, the possibility of error. We have spent our journey marveling at the fidelity of life. We have seen how DNA polymerase replicates the entire 3 billion letter genome with near-perfect accuracy. We have seen the meticulous proofreading systems and the precise choreography of transcription and translation, but near-perfect is not the same as perfect. Life is a chemical process, subject to the laws of physics and chance. The cellular machinery, for all its brilliance, can make a mistake. The DNA molecule itself, for all its stability, can be damaged by its environment. When a change in the DNA sequence occurs, and that change is not repaired and becomes permanent, it is called a mutation. In this lesson, we will explore what happens when the genetic code, the very book of life, is altered. We will define what a mutation is, explore the different ways the code can be changed, and discuss the profound consequences for the individual and for all of life. A mutation is simply a change in the nucleotide sequence of an organism's DNA. These changes can be large, or they can be vanishingly small. First, there are the large-scale chromosomal mutations. These are major structural changes that affect large segments of a chromosome or even entire chromosomes. In our last lesson, we discussed aneuploidy, like the extra copy of chromosome 21 inches Down syndrome, which is one such mutation. Other types exist. A large piece of a chromosome can be deleted, a segment can be accidentally duplicated, a piece can be flipped in a translocation, or a segment can be inverted. These are, almost always, major events with significant consequences, as they involve adding or removing hundreds or even thousands of genes at once. For this lesson, however, we will focus on the much more common, smaller scale changes. These are point mutations, which are changes that affect just one or a few base pairs. Think of them as typos in the recipe. These small changes fall into two primary categories. The first category is a substitution. This is the simplest typo. One base is accidentally swapped for another. For example, during replication, an A might be written where a G was supposed to be. This changes a single letter in a single codon. The second category includes insertions and deletions. As their names imply, an insertion is when one or more extra bases are accidentally added to the sequence. A deletion is when one or more bases are accidentally removed or lost. As we will see, these small changes of just one or two letters can have far more devastating consequences than a simple substitution. These changes can arise in two ways. They can be spontaneous mutations, which are simple errors made by DNA polymerase during replication that the proofreading system missed. Or they can be induced mutations, which are caused by external factors in the environment. These mutagenic agents, or mutagens, are things like ultraviolet, radiation from the sun, X-rays, or certain chemicals, like those found in tobacco smoke, that physically damage the DNA, causing it to break or change its chemical structure. The consequence of a point mutation depends entirely on what was changed and where it was changed. Let us look at the potential effects. First, consider the substitution. What happens when one letter in one codon is swapped? Sometimes, nothing at all. This is called a silent mutation. Remember that the genetic code is degenerate or redundant. Code CUC, CAR, CUA, and CUG all code for the amino acid leucine. If a mutation changes a CUU codon to a CUC codon, the word has changed, but its meaning has not. The ribosome still reads leucine, the correct amino acid is added, and the final protein is identical and perfectly functional. Sometimes, the change results in a missense mutation, a missed sense mutation. Here, the codon change does result in a different amino acid. A GGC glycine might become a GAC chimes, aspartic acid. The severity of this depends entirely on the situation. If the new amino acid is chemically similar, or if it is in a non-critical filler part of the protein, it might have no effect. But if it happens in the active site, the critical, functional heart of the protein, it can change its shape and break it. The classic example is sickle cell anemia. A single substitution 1A changed to a T causes one amino acid to be wrong in the hemoglobin protein. This single change is enough to cause the entire protein to misfold under certain conditions, deforming the red blood cell and causing the disease. The most severe substitution is a nonsense mutation. This is when the codon is changed from one that codes for an amino acid like UGC for cysteine to one of the three stop codons like UGA. This is a devastating error. It prematurely terminates translation. The ribosome stops in the middle of the recipe, releasing a short, unfinished, and completely non-functional protein fragment. Now, let us consider insertions and deletions. If one or two bases are added or removed, they cause what is known as a frame shift mutation. This is almost always catastrophic. The genetic code is read in a specific reading frame 3 letters, then the next 3, then the next 3. Imagine a simple sentence: the fat cat ate the rat. if we delete the first E, the entire reading frame is shifted. The cell now reads THFATCATA, tet at. The message becomes complete and utter gibberish from the point of the mutation onward. The same is true if we insert a letter, the FXATCA tat ethiera t. A frameshift mutation garbles every single codon downstream of the error, resulting in a completely different and almost certainly non-functional protein. When we hear the word mutation, we almost always think of it in a negative context: disease, cancer, defects. And it is true, most mutations that have any effect at all are either neutral or harmful. They are the root cause of thousands of genetic diseases, like cystic fibrosis, Huntington's disease, and sickle cell anemia. These are conditions where an individual inherits a broken gene from their parents. Mutations are also the fundamental cause of cancer. Cancer is not a single disease, but a family of diseases all caused by an accumulation of mutations in a single cell's DNA over a lifetime. These mutations break the cell's off switches, the tumor suppressor genes, and jam its on switches, the oncogenes, causing it to divide uncontrollably. But this negative view is only half the story. In truth, mutations are the most important engine of life. They are not an error in the system, they are the system. Mutations are the ultimate and only source of all new genetic variation. Most mutations are neutral, a few are harmful, but very, very rarely a mutation is beneficial. A random typo might slightly change a protein's shape in a way that allows it to work better in a new environment. A bacterium might gain a mutation that makes it resistant to an antibiotic. A plant might gain a mutation that allows it to survive a drought. A human, thousands of years ago, gained a mutation that allowed them to continue digesting lactose, milk, sugar, into adulthood. This variation is the raw material upon which natural selection acts. Evolution does not create new traits on demand. Mutation provides the random variation, the new ideas. Natural selection then selects the ideas that work best in a given environment. The beneficial mutations are passed on, while the harmful ones are filtered out. Without mutation, life would be static. It would never have adapted, never have diversified, never have evolved from the first simple cell into the staggering, beautiful complexity we see today. And here, our journey concludes. We have explored the foundational principles of molecular biology. We began with the search for the molecule of life, finding it in the elegant double helix of DNA. We learned to read its four-letter genetic code, written in three-letter words called codons. We marveled at the machinery of replication, the semi-conservative process that copies this vast library with such fidelity. We followed the central dogma, the flow of information from the permanent DNA vault to the temporary RNA messenger in transcription, and from that message to the final, functional protein in translation. We have defined the gene as a complete functional unit, and we have seen how the cell, with profound wisdom, regulates these genes, turning them on and off to create the symphony of life. We saw how this entire library is packaged into the dense, organized structures we call chromosomes. And finally, we have seen that this system is not static, it is alive, it changes. We have learned that mutations, these errors in the code, are not just the source of disease, but the wellspring of all variation, the engine of evolution itself. This knowledge is now a part of you. It is the fundamental story of how your own body functions, moment to moment. Let it settle. Let it integrate. As you continue to rest, your mind will organize and store this information. You have done well. Now, sleep.